So, good afternoon, viewers at home. My name is Bio-Edutainment Michael. This is another opportunity to further our discussion on microbial evolution. Today, we have come to a special instrument in evolution, and that is what is a phylogenetic tree. What is a phylogenetic tree? Don't forget that I told us about application or the use of phylogenetic tree as part of the instrument they use in classifying microorganisms. Be it microorganism or what have you, a phylogenetic tree still remain a valid instrument they use in classification. Again, the first type of method of classification is classical classification, or what we can call traditional method of classification. Thereafter, scientists thought that that was not actually suitable and not even very specific, because it was there were a lot of errors and misgivings when we only depend on the cultural method of classification by the use of um, classical method or traditional method, because that one will only rely on physical or what we can call morphological way of classification. Now the second one immediately after that was when Carl Woese actually introduced phylogenetic tree in classifying organisms into three domains. And you know the instrument, instrument he used under that was a belief that genetic tree, that all organisms must have come from the same ancestor, which was aquatic in nature. Now today, just like I said, so that we won't actually have to waste our time, a phylogenetic tree is a kind of diagrammatic representation. Is a diagrammatic representation which represents the evolutionary relationship, evolutionary relationship of different organisms and species. Different organisms and species. Don't forget that I told you about what is called diversity, and I told you that there is strength in diversity. If you pick any ecosystem, that ecosystem must have contained millions of bacteria that will coexist together, and in that kind of community, it is a community of diverse organisms or diverse species of that. So that is what a phylogenetic tree is. Anywhere you come across philo, that is talking about evolution. Evolution. So it's a kind of a tool that tend to rely on the trends, the evolutionary trends in the classifying or establishing how related organism could be from a different species or organism. So, and there are different types. So the first type is what is called rooted phylogenetic tree. Just from the name rooted, so rooted means that it has a common ancestor. For example, if you draw something like this, a tree, a phylogenetic tree that is looking like this, you will discover that irrespective of the number, irrespective if this is A, B, C, D, E, irrespective of the number of branches, you will just discover that all of them must come from the same ancestor. That is why it is called rooted. And that of unrooted, that is when there is no certainty, or nobody could actually establish whether those organisms will actually have a common ancestor or not. In terms of evolutionary relatedness, if scientists are still unsure about whether they have the same ancestor or not, what they will use is they will make use of unrooted phylogenetic tree to be able to actually establish their evolutionary relatedness. Then this third type, which is called cladogram. Cladogram. This is a type of unrooted, a kind of unrooted diagram, which has to do with evolution. This one is actually depend on evolutionary divergence. Evolutionary divergence. Nothing like a similarity, but sorry, evolutionary divergence is talking about the rate of unrelatedness, but it is unrooted. That is unrooted. Then the fourth type, um which is called the so before I go into that, and that kind of divergence does not by any means indicate the time, and when there was that kind of emergence. So there was no actual time. So the same thing with chronogram. Chronogram is a kind of, this one is rooted, unlike cladogram that is not rooted. This one is rooted, and that is also talking about the degree of divergence. So, and in terms of time, there's no specific time in terms of their evolutionary differences. Okay, so that is that about um, um, chronogram. So the last type, which is phylogram. Phylogram is a kind of branches that are proportional. The branches are proportional to the length of time. So that is, the longer they are in terms of the length, that means the longer they have diverged in terms of their evolutionary. I am not actually interested in letting us have the diagram of each of these so that we'll be able to differentiate it, just because we just want to mention the type. By the time we get to the use of bioinformatics in plotting phylogenetic tree, we are going to see more of this. Now let's now look at what are the importance. How important are phylogenetic tree to discuss and to us as a microbiologist? So the first aspect we are going to consider is what are the importance of phylogenetic tree in clinical microbiology. Don't forget that the last video they had, I think yesterday, we were able to look at how can we carry out clinical diagnosis by the use of some of the laboratory principles and laboratory techniques. So now in clinical microbiology, one of the ways that we make use of phylogenetic tree is in identification and classification. So in identification and classification, you should know that any organism, for example, now if you draw a phylogenetic tree like this, if you draw a phylogenetic tree like this, and you have, if this is a one node, this another node, this another node, this another node. What this means is that, if this is the roots, what this means is that there is a small similarity between A and B than C and D. Sorry, if this is D, if this is C, this is D. So if you are to now look for a way of making use of this type of a phylogenetic tree, in plotting or in identifying and classifying these microbes, you will discover that A and B are closely related, and that any infection that will be caused by A and B will be closely related. It's going to be more very, very closely related, and that means that you are still going to make use of the same drug in treating them as well. So we make use of phylogenetic tree in identification and classification. If you want to know how closely organisms are related, you will make use of a phylogenetic tree. And if you want to know how where they can be classified, you also make use of a phylogenetic tree. And don't forget the three domains of life. The three domains of life that has something like this. Okay, so if the was is a eukaryote, this is archaea, and this is prokaryote. So from what you will know that archaea and prokaryote, they are what? They are very, very similar than when you compare archaea with eukaryote. So that is the meaning of that. So that means if you want to know all the organisms that are in the same group under archaea, that means you begin to make use of a phylogenetic tree in identifying them. Then it is also used to understand and study antibiotic resistance. Antibiotic resistance. You should know that organisms, if you have two or three organisms that are resistant to similar group of antibiotic, you should know that you can also use this one to classify them based on their antibiotic resistance properties. So the use of phylogenetic tree also continues when you can apply it in the determination of antibiotic resistance. Then the third one is that it also helps us to be able to track transmission and rate of infection. For example, if there is an outbreak, if you want to know, the real source of real source of that kind of infection, you make use of a phylogenetic tree and you'll be able to trace it back to the. So that is why it is very, very important in this. Then the fourth application is that it is used in viral evolution. It is used in viral evolution. If you want to know how related evolution viruses are, you apply phylogenetic tree to be able to know them, and also in vaccine development. So the last one is that it is used in microbial forensic. So microbial fingerprinting, and it also has a legal application. For example, now if you now discover that you isolate similar or related microorganism criminals from two criminals, you can actually use that one to be able to detect whether the real application in the forensic microbiology. So that is the first relevance or application of phylogenetic tree in clinical microbiology. I told you it is used for identification and diagnosis. This one is it is used for identification. That means you can use it in diagnostic studies. So this one is also in diagnostic studies as well. That is antibiotic resistance. You want to know organisms that are related. That means they will develop resistance to similar, very closely related antibiotics. And if you want to establish contact tracing, if you want to establish the real origin or the real source of transmission or an infection or any outbreak, you can make use of phylogenetic to establish that. Also, viral evolution and vaccine development. Okay, so the application, what are the importance of phylogenetic tree in molecular biology? How useful is phylogenetic tree in molecular biology? The first thing is that it will help us to be able to understand evolution relatedness. Evolution relatedness, that's something you call molecular evolution. We tend to believe that okay, for example, if you, it's one of the evidence that Charles Darwin used in the support of establishing their theory of evolution. They discovered that similar organisms, related organisms, they have similar shape in their chemical composition. So that is a phylogenetic tree will actually help us to be able to understand the evolution relatedness of species. Okay, now apart from that, it is phylogenetic tree will also give us a model, will give us a model that will give us the assurance or evidence that living organisms are from the same ancestor, they are from common ancestor. Then the second application is gene function and mechanism. So what you should know is that the use of phylogenetic tree, we can use it to determine functional genes and the way they can, because when you have similar protein, for example, similar protein, okay, so you see, when you look at okay, molecular biology, abi, their application, where I said it's okay, I've said that the use in phylogenetic trees are used in understanding evolutionary relatedness. Then the second one, which is gene function, you will use gene function and mechanism studies. Okay, let us see that let us assume that you have some gene A. Okay, if this one is from species A, species B, species C and D, and there are certain genes, that are located, okay, sorry, okay, now let's assume that if this one is just 30 nucleotide base, if this one is 42, if this one is 35, if this one is 124. Now on this, at point of 15, you have a specific gene that is located there, and this is at 15. At 17 as well. You have another one. You now map this another gene at position, let me say position 33. And if there's another gene that you now have it at 105, for example, under normal circumstance, you should be able to know by the use of, because if you want to plot phylogenetic tree for this, you are going to do it this way, that 15 will first of all come, that is A, then followed by B, the gene in B which is 17, followed by the gene in C which is 33, then followed by that gene which is located on 5. So this is going to be A, B, C, D. Now, what that means is that the gene on A and B, which is on will be similar. Their function will be similar, and apart from that, they will also use their mechanism of action will also be very, very similar. Because what it means is that by the use of this phylogenetic tree now, you will know that A and B are closely related than D to A or C to B. So that is how you can make use of phylogenetic tree to be able to know the function of certain genes and their mechanism of action. The mechanism of action. So the third point under that is their usage in comparative genomics. In comparative genomics. Now, what do we mean by genomics? Genomics is the study of genome. Is the study of genome. Genome means total, the total gene, the sum total of gene in any organism is called the genome. So what it now means is that if you now have organism, if one organism is having if let's say you have one organism that is having 500, okay, let's say 5 million base pair, for example 5 million base pair, and you have one that is just 500 base pair, I mean 5,000, another one has 20,000 base pair, another one has 120,000 base pair. Now, what it means is that those ones that have, okay, let's say you now have another one which has 5.2 million base pair. So what that means is that you will be able to know, this is comparative, when you are comparing the genome of this with this one, with this organism, in order for you to identify them. That is exactly what is called comparative genomics. Comparative genomics. Or when you now pick this one, don't forget that we have when you now compare this one with another one, for example, if you use whole genome sequencing, and by the time you now look at them comparing found in A with that is found in other, so that is what we call comparative genomics. And when you now draw a phylogenetic tree that now looks like this, so you go and get, you go to the gene bank, go and get the sequences of organism A, get that of organism B, get that of organism C. So when you now start mapping them together, this is what we call molecular mapping. When you start mapping them together, you now see that maybe on their genome, on their genome, there will be some genes that will serve the same function, that will serve similar function or the same function. That is the application of what phylogenetic tree in comparative genomics. Then the next one is the usage of phylogenetic tree in determination of a protein evolution structure function relationship. Now, when you compare mitochondria, compare this. Okay, this is going to be a very good example of how you can use phylogenetic tree knowing the function of certain protein. Now let's, for example, in plants now and in animal, you have chloroplasts in plants and you have mitochondria in animal. So when you look at the shape of the two, they are very very similar. If you look at the shape of mitochondria in animal and you look at the same shape in chloroplast, you will discover that they are very similar, and that is telling you that they must have come from common ancestor. So the same way, what you can use in you can use phylogenetic tree to do. Okay, so the last one, okay, the next one is development of molecular markers. What are molecular markers? Molecular markers are just like each, they are the identity card. uh, they discover that on the surface of some chromosome, on the surface of DNA, there are molecular markers that you can use to be able to identify them. It's just like a signboard. It's just like a signboard. And there are some cells as well that they have potential to develop into cancer in the future. So these are kind of what you call molecular markers, and you'll be able to show. So organism, if you are doing that from phylogenetic tree, so if this is A, B, C, then this is D, this could be E, this could be F. Now looking at ABC here, ABC here, what this one is telling you, this phylogenetic tree is telling you, is that because we have ABC on the same branch, although the time of their divergence actually differ, but because they are on the same branch, that means, and when you now check the molecular marker is just like a signature. That's why I said it is an identity card, something that identifies that, oh, anybody that sees it will say, and all these organisms, they have their own molecular markers as well. The molecular markers of Salmonella typhi is quite different from that one of Shigella. It's quite different from other bacteria as well. So because if you now want to draw phylogenetic tree, you now see that anyone that are related, they will tend to have this very closely related molecular markers. And the last point that we are going to discuss today, and that is gene and genomic sequencing project. You know, I told you it is this one, the phylogenetic tree, will help us to be able to understand you should know that you should be able to understand the relatedness between what is called genus, species, family, that is a taxonomic hierarchy. So you now discover that phylogenetic tree is going to help us to be able to understand this one as well. And that is what they use in sequencing them. They use phylogenetic tree. When you draw a phylogenetic tree like this, let's say this is the phylogenetic tree, and you want to consider, you want to sequence the genome of A, that of B, you collect DNA of A, you collect that of B, you collect that of C, you collect that of D, you collect that of E, you collect that of F. So you now go and sequence them. By the time you do what is called whole genome sequencing, you'll be able to know how closely related they are, and how closely unrelated they are. And you will now be able to compare them with a standard which is called the reference train. Every microorganism has its own reference. If you have, okay, if you are talking of E. coli has its own reference. Even if you do not know what you have isolated, you just know that the same, they contain the same or very similar closely related numbers of sequences as you have it there. So just like I said, that today's lecture is actually devoted for understanding what phylogenetic tree is, how important it is in molecular biology, and how important it is to clinical microbiology, for diagnosis, for treatment, for development of drugs and some others. And I've also mentioned different types of phylogenetic tree, which include rooted, unrooted, cladogram, clonogram, as well as the last one, which is the phylogram. So